आप सुन रहे हैं सुनो किताब।
Class 11 Biology Chapter 3 Plant Kingdom Part 1. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker, wherein he suggested the five kingdom classification with Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae through earlier classifications, place them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe Plantae under algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Let us also look at a classification within angiosperm to understand some of the concerns that influence the classification systems. The earlier system of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves. They were based mainly on vegetative characters or on the androecium structure. Such systems were artificial. They separated the closely related species since they were based on few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed which were based on natural affinities among the organism and consider not only the external features but also the internal features like ultrastructure, anatomy, embryology, phytochemistry. Such classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have common ancestor. We now use the information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituents of the plants to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing, simple, thalloid autotrophic and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and woods. Some of them also occur in association with fungi and animals. The form and size of algae is highly variable. The size ranges from microscopic unicellular forms like Chlamydomonas to colonial forms like volvox and to filamentous forms like Ulothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being zoospores. They are flagellated and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size or non-flagellated but similar in size. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in some species of Chlamydomonas, is termed as an isogamous. Fusion between one large, non-motile female gamete and a smaller, motile male gamete is termed as oogamous. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary procedures of energy-rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, for example algin and carrageen, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella and Spirulina are unicellular algae rich in proteins and are used as food supplements even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in structure. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eurothrex, Spirogyra, and Cara. 3.1.2 Phaeophyceae. The members of the Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous form ectocarpus to profusely branched form as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminin or mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside of a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the oogonium. The gametes are pyriform and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. 3.1.3 Rhodophyceae. The members of the Rhodophyceae are commonly called red algae because of the predominance of the red pigment R-phycoerythrin in the body. Majority of the red algae are marine with greater concentration found in warmer areas. They occur in both well-lighted regions close to surface of water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Galledium. 3.2 Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly grown in moist, shaded areas in the hill. Bryophytes are also called the amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks soil. The plant body of bryophyte is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organ in bryophyte are multicellular. The male sex organ is called antheridium. They produce biflagellate anthrazoids. The female sex organ are called archegonium is flask-shaped and produces a single egg. The anthrazoids are released into water where they come in contact with archegonium and anthrazoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichen, are the first organism to colonize rocks and hence are of great ecological importance. They decompose rocks. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is a thalloid, for example Mackentia. The thallus is dorsiventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts take place by fragmentation of thalli or by the formation of a specialized structure called gemme. Gemme are green, multicellular asexual buds which develop in small receptacles called gemma cups located on the thalli. The gamete become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. This sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is the leafy stage, which develops up from the secondary protonema as lateral bud. They consist of upright, slender axis-bearing spirally arranged leaves. They are attached to the soil through the multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs, antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spores dispersal. Common examples of mosses are Funaria, Polytrichium, and Sphagnum. 3.3 Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionary, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places though some may flourish well in sandy soil conditions. You may recall in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophyte, the main plant body is the sporophyte, which is differentiated into true root, stems, and leaves. These organs possess well-differentiated vascular tissues. The leaves in Pteridophyta are small, microphyls as in Selenginella or large macrophyls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct, compact structures called strobili or cones. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small, but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophyte bear male and female sex organ called antheridia and archegonia, respectively. Water is required for the transfer of antherizoids, the male gametes released from the antheridia to the mouth of archegonium. Fusion of male gamete, therefore, with the egg present in the archegonium result in the formation of zygote, which is the dominant phase of pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selenginella, Salviana, which produce two kinds of spores, macro and microspores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of the zygote into young embryos takes place within the female gametophytes. This event is precursor to the seed habit, considered as an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Lycopsida, Phoenospida, and Pterospopacida. 3.4 Gymnosperms. The gymnosperms are plants in which the ovules are not enclosed by any ovary and remain exposed, both before and after fertilization. The seeds that develop the gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne sporophylls which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili-bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined only to a limited number of cells. This reduced gametophyte is called a pollen grain. This development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree. However, in cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure called an ovule. The ovules are borne on megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides to form meiotically to form 4 megaspores. One of them, megaspores, enclosed within the megasporangium, develops into multicellular female gametophyte that bears 2 or more archegonia of female sex organs. The multicellular female gametophyte is also retained with megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Unlike the gymnosperm, where the ovules are naked, in the angiosperms or flowering plant, the pollen grains and ovules are developed in specialized structure called flowers. 3.5 Angiosperms. In angiosperms, the seeds are enclosed by fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from tiny, almost microscopic wolfia to tall trees of eucalyptus. They provide us with food, fodder, fuel, medicines, and several other commodities that are commercially important. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized with having two cotyledons in their seeds, while the monocotyledons have only one. The male sex organ in a flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. The anthers following meiosis produce pollen grain. Female sex organ in a flower is the pistil or the carpel. Pistil consists of an ovary enclosing one to many ovules. With ovules are present highly reduced female gametophytes termed embryo sacs. The embryo sac formation is preceded by meiosis. Hence, each of the cells of an embryo sac is haploid. Each embryo sac has 3-celled egg apparatus, 1 egg cell and 2 synergists, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. 3.5 Angiosperms. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where two male gametes are discharged. One of the male gametes fuse with the egg cell to form a zygote. The other male gamete fuse with the diploid secondary nucleus to produce the triploid primary endosperm nucleus. Because of the involvement of two fusions, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo and the pen develops into endosperm, which provides nourishment to the developing embryo. The synergists and antipodals degenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm can be shown diagrammatically. 3.6 Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide to mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gametes producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. First, sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae, such as volvox, Spirogyra, and some species of Chlamydomonas, represent this pattern. Second, on the other extreme is the type wherein the diploid sporophyte is the dominant, photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to a few celled haploid gametophyte. This kind of life cycle is termed as diplontic. All seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern. Third, bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in their dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternates with the short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic, vascular plant body. It alternates with multicellular, saprophytic, autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus and Alga is diplontic. Summary: Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperm and angiosperms. Algae are chlorophyll-bearing, simple, thalloid, autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely Chlorophyceae, Phaeophyceae, and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axis-bearing spirally arranged leaves. The main plant body of a bryophyte is gamete-producing and is called a gametophyte. It bears the male sex organ called antheria and female sex organs called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The sporophyte bears sporangia, which produces spores. The spores germinate to form gametophytes, which require cool, damp places to grow. The gametophyte bear male and female sex organ called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces sporophyte. The gymnosperm are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked-seeded plants. The gymnosperm produces microspores and megaspores, which are produced in microsporangia and megasporangia, borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on the axis to form male and female cones, respectively. The pollen grain germinates and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organ stamen and female sex organ pistil are borne in flower. Each stamen consists of a filament and an anther. The anther produces pollen grains after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with egg cell, syngamy, and other fuses with the diploid secondary nucleus, triple fusion. This phenomena of two fusion is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generation between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycle, haplontic, diplontic, or intermediate.
सुनो किताब से सुनने के लिए आपका धन्यवाद।